Okay, continuing on. Um, I talked at the very end of the last lecture that mitosis produces two identical diploid daughter cells. Meiosis produces four haploid daughter cells, which means not only do they have half the amount of DNA that the cell they came from would have, but also that DNA can be a little bit different because, well, every cell produced by meiosis has a complete set. It has one complete set rather than two of the DNA in the cell. They may have different alleles of certain genes. So for example, if someone has the dominant capital A allele on one chromosome and the recessive little a allele on another chromosome, some of the gametes they make will have big A, others will have little a. And that's an important thing to remember. Another thing that meiosis does is that homologous chromosomes, that is the two chromosomes that are parts of a pair, and you can see this here where you have the blue chromosome and you have its partner, the red chromosome. They have lined up with each other and they have got what is called a synaptonemal cortex, where they have kind of joined up with each other and they can actually swap pieces. And these pieces will contain the same genes, but not necessarily the same alleles of those genes. So again, the main point of this is it increases the genetic variability. Later on in a later chapter, you will see how multiple genes are found on a given chromosome. And so you have what are called linked genes. And what this does is this allows a little more variation in with those linked genes. So this is just showing this in a little bit more detail. On the left-hand side of your screen, it's showing crossing over in meiosis 1. On the right-hand screen, it's showing what happens in mitosis. Notice in mitosis, you don't have the chromosomes paired up. You have just the sister chromatids. In meiosis, the homologous chromosomes do pair up. And so in meiosis 1, the separation that's happening is actually you are separating the homologous pairs. So meiosis actually has two stages. The first stage is called reduction division. This is what goes from diploid to haploid. The second stage is called equational division. This divides up the sister chromatids. On a very superficial level, each of these stages kind of resembles mitosis. But remember, meiosis is not mitosis. So in meiosis 1, you have crossing over happening in synapsis. This is where the bits of chromosomes are exchanged between members of a homologous pair. The homologous pairs line up, so you would see that in metaphase 1 of meiosis 1. Then they separate. That's anaphase 1 of meiosis 1. And at the end, you have two haploid cells, but in each of those haploid cells, each chromosome still consists of sister chromatids. Then meiosis 2 divides those sister chromatids up so that you wind up with 4 haploid cells originating from the original diploid cell. So this kind of goes through the process. You can see the diagrams here. If you have your textbook with you, you can look in your textbook. Prophase 1, the big thing in prophase 1 of meiosis is you do have this crossing over happen. Here's metaphase 1 of meiosis. Notice the sister chromatids are still there. Notice that the homologous pairs are joined up. The homologous pairs are what's going to separate. You've still got kinetochores. You've still got a spindle. Anaphase 1, homologous chromosomes have separated. Telophase 1, new nuclei are starting to form. And now you enter stage 2. Here you have 2 haploid cells that have developed from the original diploid cell. Notice in each one, there's one member of the pair of sister chromatids, one member of the homologous pair, one member of the homologous pair. Each homologous pair consists of sister chromatids. Then in metaphase 1, this really is where it superficially looks like mitosis. You've got the chromosomes lined up in the center of the cell, it's getting ready to separate those sister chromatids. Anaphase 1 or anaphase 2, sister chromatids separate. Telophase 2, now you've got 4 separate cells that form. So this next slide contrasts the two. Notice that it's showing meiosis 1, which is where the homologous pairs divide up on the top, versus mitosis, which doesn't involve homologous pairs joining up or separating at all, in the bottom. So meiosis 1 versus mitosis. And then this is meiosis 2. And of course, mitosis has only one set of stages. It doesn't have a part 2. So in meiosis 2, you're going to have here the sister chromatids splitting up in cells that are already haploid. So all of this, the whole point of this, is to ensure genetic variation. You get independent assortment of the chromosomes, you get crossing over, which you've already seen, you get random fertilization. I'm going to explain in the next segment I talk about independent assortment of chromosomes and a little bit about random fertilization.